There are many diseases and conditions that are the leading cause of death here in Canada, such as Alzheimer's, cancer, and many heart diseases. The fast-paced search for finding cures has been going on for decades, and the search is finding much hope now. The start of CRISPR began in the late 1980s and caused much curiosity as to how diseases can be cured for the long run using this technology. But major questions remained. Is this ethical? Is this safe? Is there any future hope in this technology being used for commercial use in the future? Well, we are here today to talk to you about CRISPR technology and all your ethical concerns along with it. CRISPR stands for Clusters of Regularly Interspaced Short Palindromic Repeats. CRISPR is one of the most incredible technological advancements in the biotech area. It has the ability to edit genomes by altering and editing DNA and gene functions. Its applications are to treat diseases and other harmful disorders, as well as improve the quality of crops. A protein called Cas9, or CRISPR-associated, acts as scissors and cuts the strands of DNA, just like helicase in DNA replication when it cuts the two strands of DNA apart for transfer. So how does CRISPR work? Well, there's two different types of CRISPR, one that is natural and the other one that is engineered. We're going to be focusing on the engineered process as it is still an ongoing advancement and has many issues underlying it as well, which we will mention later in the presentation. There are two players in this process, the first one being CRISPR RNA. In CRISPR RNA, there is a spacer, which is a non-coding region in the DNA that is used to regulate the process of transcription from CRISPR DNA, which is a DNA sequence found in prokaryotes that is a part of bacteriophage that has already been infected in a prokaryote, to CRISPR RNA, which can then be broken down and manipulated. The second player in this process is Cas9 protein. As mentioned before, it breaks apart two foreign DNA strands, not the CRISPR DNA. The protein then binds to two RNA molecules, the CRRNA and another called guided RNA. The two then guide the Cas9 protein to the target site of foreign DNA where it will make its cut into a double-stranded break, which is a terminology made according to a 2014 Science study. The DNA is actually complementary to the 20-nucleotide CRRNA, which is made to complement it. In general, the Cas9 protein is brought into the matching genome section by the guide RNA and the CRRNA. Then, the double-stranded DNA unwinds as shown here and gives space for the Cas9 protein to bind. Then in the second step, the Cas9 protein will break apart the DNA as shown here to make space for the new programmed DNA as shown here, similarly to how RNA polymerase will fix an unbinded spot and create a new section and bind loose ends. To show you how CRISPR actually works on a microscopic level, we will now show you a quick animation showing how the DNA is unbinded. CRISPR technology was adapted from the natural defense mechanisms of bacteria and archaea. These organisms use CRISPR-derived RNA and various Cas proteins, such as Cas9, to stop attacks by viruses and other foreign bodies. They do so primarily by chopping up and destroying the DNA of the foreign invader. When these components are transferred into other and more complex organisms, it allows for the manipulation of genes or editing. There is a secondary type of CRISPR, which is natural in bacteria and archaea, and is what the biotech of CRISPR mainly was derived from. It behaves somewhat differently than the biotech version, but its main plan comes from the natural type. To summarize, the natural process starts with the bacteriophage that has infected the prokaryote. Then the cell uses its natural CRISPR-Cas system to immune itself from the same type of virus, which is actually done by inserting a small amount of the virus into CRISPR sequence and to have these spacers near them remember this virus
like a library of some sort.
The second mutation that CRISPR can fix is cystic fibrosis, which is caused by a nonsense mutation by base pair deletion. To show you how it works, this is what your regular CFTR chain looks like. These are your CFTR enzymes that help the chlorine and salt ions go through the chain, and it goes on the outside of the cell. But when you have cystic fibrosis, these cells cannot go through or limited go through due to this mucus that builds up because there's less CFTR enzymes. Now, overall, the mutation is caused by a deletion of 3 of the nucleotides, so a codon, and the codon is then replaced by one of the stop codons, so UAA, UGA, and UAG, and then it stops production of the CFTR enzyme. Now, currently, there has been no main trials done for cystic fibrosis, but many have been taking place, and there has been some success, but the problem runs into when the cystic fibrosis causing mutation has about 1,700 different mutations, and it is hard to track down which one needs to be tackled. So far there's been no human or any other trial, but works are definitely underway in the future.
In sickle cell anemia, a single base pair is changed to now code for a different amino acid, and overall changing the protein that makes up hemoglobin. This then changes the shape of the red blood cells and causes problems such as blood clotting, organ function problems, and early death. This can be fixed through CRISPR technology. So here I have an example of what happens with sickle cell anemia. So basically, this is the normal RNA sequence where this codes for this amino acid, but instead, when there is a mutation, it codes for this one. So it's the change in the missense mutation. What CRISPR technology will now do instead is it'll remove the part of the DNA that changes the amino acid sequence and instead insert a programmed DNA that'll code for fetal hemoglobin, hoping to replace the sickle hemoglobin. This treatment has already been done to a 30-year-old woman from California, and within a year later, she is still doing well after the modifications. She has said that she feels much healthier now and does not face any severe side effects. Through tests, it is shown that 99.9% of her red blood cells contain fetal hemoglobin, keeping her healthy now.
So this is our debate section of the presentation. We are going to be talking about the pros and cons of CRISPR. I am the pros and Animal is the cons. So to start off on the pros, we're going to look at how cancer has been taken apart into its process and how certain genes have been turned off so that cancer cannot continue growing, which is very revolutionary. And although this is not being put into commercial use yet, there have been a few trials done and it has been a very great success so far.
On the con side of things, there was a Chinese scientist in 2018 who performed the CRISPR tech procedure on 2 fertilized embryos. Now, what he was trying to do was remove the HIV gene from both of them so that they wouldn't get HIV in the future. But this was very unethical because he did not consider the babies that he was tampering with, and it was actually later discovered that he kind of forced the mother to undergo this procedure against her own will.
Well, another pro that could be taken from this is that the cost. So, although the cost is quite cheap, and especially with Canadian universal healthcare, this cost can actually be diminished to even free, and many other families can afford this process.
Gene editing may be cheap, but only for some. What is considered cheap to some people may be too expensive for others. This means that the people that would want to do this procedure and have CRISPR may not be able to afford it necessarily.
So my final point for the pro side is regarding scientists and the ethics of having cosmetology done. And a lot of scientists are agreeing that for cosmetic purposes it will be very limited because the main goal is to fix deadly diseases and conditions. Also, for anyone who has any religious beliefs against CRISPR, are not forced or told to do CRISPR, and it's all their choice. And hopefully, CRISPR will bring everyone together to realize how revolutionary gene editing is.
Though CRISPR is in its early stages, and scientists are very wary of the fact that they will not be doing cosmetic editing, we don't know in the future if this will be the case. And if, say, this is the case and children are born with cosmetic editing where they have blue eyes or brown eyes or they are built a certain way because their parents wanted them to, this could lead to segregation within children. Because if someone went through CRISPR and had cosmetic editing done, the other children that did not may feel lesser and may feel cheaper in a way.
And that concludes our debate on the pros and cons of CRISPR. I hope that you learned something from both sides and now you have a better understanding and you know which side you agree with.
Thank you.
Thank you.
One of the current developments in CRISPR and Cas9
Is new ways to edit the genes. One of them being taking out only a single nucleotide, which means one letter of a codon, from the gene to code for something different, as well as copy and paste different genes in the same DNA to turn on and off specific gene functions.
Many other smaller projects are being tested using CRISPR technology, such as editing chocolate to remove harmful fungi, altering unhealthy human fat into healthy human fat, creating genetically modified mosquitoes to either lower the overall population or completely remove it as a whole by inserting the Cas9 protein into its germline so that its other generations can also be affected, curing blindness in humans, Huntington's disease, and many other more smaller projects.
In the start of October 2020, two scientists named Miss Emanuela and Miss Deduna both earned the Chemistry Nobel Peace Prize regarding CRISPR. They found a new protein complex that also helps guide the Cas9 protein called tracer RNA. This was discovered through a bacteria called Streptococcus pyogenes, which is found in many conditions, lung conditions such as tonsillitis. What they did was they discovered that although the guide RNA and cRNA both guide the Cas9 protein to the DNA, many other smaller proteins also help it, one of the more active ones being tracer RNA. This experiment was copied into a test tube and then simplified for the CRISPR biotech that we need.